Okay, well, thank you very much for the opportunity to talk to everybody today. Dallas and Sask Wheat, appreciate it very much. Much as I do appreciate this great technology, I am looking forward to the day when we can do this in person again. And you don't have to hear the phrase, you're on mute, you're on mute. Okay, so I'm going to talk about Fusarium head blight. Talk about mostly the work that we've been doing in my lab with a lot of great people. I work with some wonderful graduate students, some postdoctoral fellows, and we always have some good summer students. So at least this year, our summer students are allowed to come in and not work from home as much as they did last year. So grateful for that. So I'm going to assume that most of you on the call or watching this later have a pretty good idea what Fusarium head blight is. I'm not going to spend a lot of time on that, but I will touch on a couple of interesting things we found with the pathogen. I want to talk kind of briefly on cultural control, really the crop sequence study that we have. The data is a bit preliminary, but we're finding some interesting things. And then move on to some of the work we've done in genetic resistance, particularly looking for new sources of resistance. And the last part of the talk, and the longer part, will be the Fusarium fungicide management strategies. So timing and how we can try and improve what we're doing for managing Fusarium. So it's really an integrated pest management program. So you're going to hear me repeat this a few times. There is no one thing we can do to manage this disease well. We cannot spray our way out of it. Unfortunately, genetic resistance is improving in the hard red springs especially, but lots of other classes, there's not a lot of resistance there. And I think we could do a better job of sequencing as long as you folks can figure out how to make it profitable. So what is the disease? It's a disease of cereals, corn, I'll mention, and other grasses. And it's the same Fusarium species that cause Fusarium head blight. Many of them you'll find on the seed or as seedling blights or stem or root rots. So there's quite a list of them. I've listed 5 of the more, I guess, common ones we see with Fusarium head blight. If you're good and you take a lot of practice, you can actually look under the microscope and identify them from the morphology of the spores or the culture on the plate. And then, of course, you could use PCR diagnostic or molecular diagnostic tests. So these are quite common ones. You've probably heard of most of these by now, but the one we're going to focus on is Graminearum. It's the one that gets most of the blame. And so we'll talk a little more in depth on that one. So these are the symptoms you're likely to see. I'm sure many of you have seen probably the end of July or so, beginning of August, you start to see premature bleaching of those spikes. So what's happening or what has happened is that the spores of the fungus have got into a floret and they've infected a spikelet, and then the pathogen will grow up the rachis. And so it can grow to the tip here. You see it's nice and green down below, and yet it's already bleached here, faded. This one, it hasn't gone all the way to the top, but you see some damage in these kernels. Or on the right, pretty much the whole spike has been affected prematurely, so it's ripened. So when we're out rating for Fusarium head blight, we have about a week roughly, sometimes not even, to really have ideal conditions to see these kind of symptoms before everything becomes bleached and ripened. Under some conditions, you might actually see this. You might see very orange sporulation on the glooms, and that's an indication of the pathogen is sporulating, and kind of a pretty color, I think, but probably not if you're wanting to grow wheat. For us pathologists, we're always amazed at the colors and shapes and things that fungi form. But as a result, you end up with Fusarium damaged kernels. So you get a sample that looks like this. You get more than 4% FDKs, Fusarium damaged kernels, or tombstone kernels. You're looking at sample grade. 10%, you're looking at salvage. So that affects yield, obviously. It also affects protein content because the chalky kernels generally have very low protein content. And that's what the grain is being bought at is the amount of damage to those kernels. And then also the amount of toxin in the sample. So kind of a double whammy, yield, quality, parameters. So what's happening with the disease is that the pathogen is overwintering on the stubble of the previous crops. And on that stubble, you're getting ascospores produced. or maybe macroconidia. So the ascospores are the sexual stage, you're getting some genetic variability of the pathogen, and the macros or macroconidia are the clonal spores. So masses of them are produced, they get up onto the spikes during flowering. So our winter wheat, maybe next week, we'll start to see flowering already, and we may have some infection, especially if we have conditions like we're getting this week with a bit of rain. A little bit warmer would probably be better for the fungus. And 2 or 3 weeks from now, when the spring wheat is flowering, those spores are going to get up on the heads. We also know that it's not just on the past wheat stubble, it's many different crops, right? Barley, canary seed even, corn moving in from the east. The pathogens tend to do pretty well on some of these crops. So those spores may not necessarily being produced just on the last wheat crop, but on many of the other crops that are being grown in the rotation. So our strategy in my program is to look at an integrated pest management program. So we're looking at residue management really in terms of the crop sequence. So can we keep those crops that produce a lot of these spores, the residue of those crops limited, or the time period between those susceptible crops lengthened so that we don't have as much residue to deal with. The second part of the talk, we'll talk a little more about genetic resistance and how we're looking for some new sources and how we find those sources. And then finally, we'll end off with fungicides and what we might be able to do better. Okay, I want to talk about Gursahib Singh's work. Gursahib just finished his PhD with me in April, so he is now Dr. Singh, and Gursahib is out at Outlook. He's working with Gary Nadowich, and he's, I think, really enjoying it, although he mentioned the other day he's noticing that being a student now seems easy. He only had 4 projects, now he has 65. So he's a really busy guy. Anyway, what Gursahib did was to look at the amount of Fusarium DNA in these wheat samples that we collected. I don't know if my cursor is working here, in 2014 to 16. So if you remember, those years were pretty bad Fusarium years, especially 2016. 15, not so much, but 14 and 16, we had a lot of problems in the province. So these were samples that Amy Brown gave us from Sask Crop Insurance. We don't know anything about the growers or anything, but we do know whether the samples are Durham or hard red, and that they were sent in because there was serious Fusarium problems, Fusarium headlight damage. So when Gursahib looked at those, what he found, as we expected, was Graminearum is the main culprit. So we have about 4 times as much Graminearum as the next most common species, which is Abenaceum, and about 10 times as much as Calmorum. I think it's about 40 times as much Bowie and 65 times as much Torquioides. So obviously Graminearum is the main pathogen that we're worried about, and that's reflected in those samples that were collected in those bad years that we had for Fusarium. Okay, this is a pretty busy slide, so I just want to give you a brief idea of, you've, I'm sure, heard of DON. You must have. You may have heard of the 2 different forms, 3-ADON, 15-ADON, and there are a number of other toxins we talk about that Fusarium graminearum produces. Other Fusarium species as well produce different toxins. There's one called NX, so we'll talk a little bit about that in the next slide. I'll introduce it, and another one called NIV. So there's kind of 4 that I'm going to talk about: 3-ADON, 15-ADON, which end up being turned into DON, and these other ones called NIV and NX, or NX2, which turns into NX. So they all start with a common molecule up here. I think it's calonectrin it's called. If you have this tri1 gene, one form of it, you end up with the 3 and 15-ADON forms. If you have another form of that gene, or another gene sequence for tri1 gene, you end up with NX2 here. 3A NX means NX2, and NX. And then these different chemotypes we see, 15-ADON and 3-ADON, it's really this acyl group. So it's got a carbon here, a double bond to the oxygen, and a carbon at the end. It's at this 3 position, as opposed to the 15, where it's not there at the 3 position, but it's there at the 15 position. And if you knock them off at both positions, you end up with DON, right? So it's just different forms of very closely, very similar molecules, what I'm trying to say. But basically, we're seeing differences in the pathogen depending on how much of these 3-ADON, 15-ADON, NX, NIV chemotypes of the pathogen are present. So just for a little history, 20, 25, 30 years ago, what we had was mainly all 15 ADON chemotypes of Fusarium graminearum in Saskatchewan. So if you remember, we probably didn't have a lot of Fusarium problems at that time, but from harvest samples and surveys, certainly Fusarium headlight was detected. I remember seeing it in the mid-90s on crops up in Melford when I first started up there. Nobody worried about it. It was very minimal. It was not on anyone's radar, to my knowledge, but it was there. And so we had 15 ADON chemotypes. Now, if you look at the samples that Gersie analyzed from that 2014 to 2016 period, you find that that's changed, and that two-thirds of them now are 3 ADON. So why is that important? Well, we'll take a look at that in a minute. We were also able to identify from those samples how many were durum. So actually most of them were durum. The durum growers in 2016 had a large problem, and you can see that 3/4 of the isolates or samples collected were 3 ADON in that sample collection on the durum. Whereas in the spring wheat is about half and half, half 3 ADON, half 15 ADON. So why is that important? We'll just look at some of the things that Gersie was able to figure out. He inoculated these plants in the greenhouse with isolates that were either 3 ADON, 15 ADON, NIV, or NX2. So ones that produce NIV or NX2 are not nearly as common, and you don't find as much toxin with them either, but they do exist. They could become more problematic. But what we're finding is that what Gersie found was that the 3 ADON isolates produce much more disease severity, higher disease severity in the greenhouse when he inoculates them than 15 ADON or the NIV or NX2 chemotypes. He also finds that mycelial growth is greater. So he did it at 3 days. So he measured this on agar. He put the isolate little spot on the plate, and then 3 days later, you come back and measure the diameter of the growth. And you find that 3 ADON isolates grow faster than 15 or the NIV or NX2 at 3 days, and somewhat similar at 7 days. You don't see the difference between 15 NIV and NX2 at 7 days, but the 3 ADON grows faster as well. If you look at the amount of spores, so that's those macroconidia I was talking about, macroconidia. So the 3 ADON is producing a lot more spores than the 15 ADON or the NIV or NX2 as well. And lastly, Gersie looked at mycotoxins. So the 3 ADON produce a lot more mycotoxins than either the 15 ADON, the NIV, or the NX2. So maybe this helps us to explain or understand why we're having more problems with Fusarium head blight in the province. Our pathogen population has changed to a more aggressive chemotype, the 3 ADON chemotype, it seems, particularly for the durum growers who have had a lot of the problems in 2016. Anyway, there was a lot of poor samples that year. So this is kind of interesting, not directly applicable to most folks' situations, but it does help to explain how come we're having more and more problems with Fusarium head blight as compared to maybe 10 years ago. Okay, so what can we do about Fusarium head blight to manage it? I'm a big proponent of diverse crop rotation. Sometimes I think my students get sick of hearing me talk about crop rotation and crop sequencing, but I think be it one of the oldest technologies to control plant diseases there is, it's still very effective for many diseases, and even for Fusarium head blight, where it's not the complete answer, it is part of the answer. So Alejandra Oviedo is a student with me doing her master's degree, and she's looking at crop sequencing impacts on Fusarium head blight and other diseases at a number of locations. So I like this shot. This is from Farming Smarter down at Lethbridge, and they just happened to kind of speed things up. They had year 1, which is this big study here, and year 2, which is on the right over here together. In the first year, they seeded 9 crops in long strips here, as you can see on the left. And so this would be 1 replicate. If you can see my cursor, hopefully you can see my cursor. And there's 4 replicates. And then in year 2, they come back and seed narrower strips of the same crops. And so you end up with 9 times 9, 81 different crop stubble effects if you consider the 2 years. So 2018 would have been the first year, 2019 the second year. And then in the third year, they come back and they seed the whole area to durum wheat at Lethbridge. We did durum at Saskatoon here, and then other locations are doing barley or spring wheat. So Alejandra's been busy now. She's in year 3. So she's just actually finished up running a lot of the toxin analysis just the last few weeks. So I think she has pretty much all her data for the first cycle of this study. So experiment 1 at all the locations. And I want to give a plug here to the CAP program, so the Canadian Agricultural Partners program, and the industry groups that funded that, including Sask Wheat here, WGRF, pretty much everybody involved with many of the cereal crops are contributing to this study. So as I said, we have 6 locations. So Lethbridge is durum, Saskatoon is durum, and Brandon is spring wheat. And then up at Melford, Indian Head, and Morden, we're doing barley. And so that what I mean by that is in the 3rd year, these plots are all seeded to one of those crops, either durum, spring wheat, or barley. All of the sites are doing at least this core collection of crops. So at least 5 different crops. And then almost everybody's doing a 6th. Everybody is doing a 6th. So what's relevant for their area? So Morden, Brandon, and I think Indian Head too all have soybeans. And Indian Head actually went all out. They've got about, I think, 10 crops in total. So they've got things like quinoa and hemp as well. Saskatoon, we have 9 crops up here. We did lentil, and Melford also did lentil. We've been finding some interesting things on lentil stubble. We also did canary seed, flax, and oat at Saskatoon. And Lethbridge did much more than 5 as well. They did about 9, I think it was, so similar to Indian Head. I think they also have quinoa at Lethbridge, if I remember all the protocol correctly. Okay, so this is fairly preliminary. We're still, Alejandra is still crunching the data, looking at what happens, but I'll just show you a few things. So at durum wheat at Saskatoon last year. So this is the wheat that we harvested, durum wheat, and then we looked at the crop it was growing on in 2019. So something kind of interesting is that we didn't see a lot of effect of the 2018 crop year. And if you remember, it was quite dry that year. So that probably explains why we're not seeing a lot of Fusarium effects from 2 years ago. But I'm just showing you, what's happened to the durum from the crop stubble it was growing on in 2019. So something interesting was that the canary seed stubble actually resulted in a fair bit of Fusarium species on the durum wheat kernels. So after harvest, we plate those kernels out and identify what's on them. And you can see if we consider all of the species on them, not just Graminearum, although probably most would be Graminearum, we find that actually surprisingly canary seed stubble, we're finding a little higher levels of Fusarium. Also durum on durum, fairly high, which is not unexpected. And even on flax, it was fairly high. So we're finding 3% of the kernels are infected with Fusarium on those crop stubbles, whereas on canola stubble, the amount of Fusarium is quite a bit less. We also had an impact of flagleaf diseases. It's pretty hard to isolate Fusarium from flagleaf diseases. So you also see that when you grow a crop on its own stubble, like durum, you get fairly high flagleaf disease severity. And that results probably in the lowest, or one of the lower yields being on durum. So durum on durum, lower yield, no real surprise there, but it is associated with leaf spots and Fusarium head blight. So we'll have some other interesting conclusions, I hope, in the next few months for the first round. And now we're repeating this study, a second round, experiment 2 at all the sites. Last year was the first year of experiment 2. This is the second, and next year will be the third, except at a few sites where COVID kind of mucked things up. So we'll probably have to extend this study by 1 year. Just something interesting at Indian Head, where we harvested barley, or Bill May and his crew did, down at Indian Head. And when we played out the samples again, we find again that canary seed seemed to have a lot of, or barley growing on canary seed stubble seems to have quite a high kernel infection. And if you look at which species it is, it's almost all POE down at Indian Head. So that was interesting and quite unexpected. So 4% of the kernels were infected, and over 3.5% were Fusarium POE. So that was a little unexpected. I would have thought Graminearum would have been the main pathogen, but at Indian Head, that's not the case. You also see on quinoa, there's a bit of a bump. So that's something interesting as well. So lots to decipher from this study. Okay, I want to move on to talk a little bit more about, talk about some of the things we've done looking at resistance. Kutcher and Brar, lots of you folks probably know Kutcher if you're reading research on wheat diseases. So Kutcher was a PhD student with us a few years ago. He's done wonderfully. He's now a professor at UBC and he's operating his own lab, and he continues to be in love with wheat. So he's continuing to work on wheat and plant diseases. So one of the things that Kutcher did was to look at type 2 resistance. So that's when that pathogen grows up the spike after it infects the florets. So he had a project where he was over at the synchrotron, and he wanted to figure out, okay, how is the resistance being manifest? What is actually causing the resistance? So he took over samples. Here he's infected a resistant variety. He's infected one of the florets, and you can see there's pretty good type 2 resistance. It hasn't grown up the rachis to the other florets. Whereas in the susceptible variety, same thing, he inoculated one floret here, and you can see it's moved up the rachis. It's much more bleached, and the kernels are going to be affected in that spike much more on the top part of the spike than the lower part of the spike. So why does that happen? So what he did, very briefly, and he did a lot of things here, I'm just giving you a very simple taste. He looked at these regions of interest, he calls them ROIs. And so he looked at the internode above, he looked at the node region. So this is this area here, this is the spikelet, this is the kernel forming just in the top left-hand area, whereas this is the rachis continuing on up the spike and below the spike. And so he looked at these three regions of interest, sort of right at the spike, the node, and above and below. And what he was able to show was really this is where the resistance is happening. This is something's changing there, whether it's lignification, whether it's a change in composition between a resistant and a susceptible variety. So this seems to be where the resistance is expressed. So Kutcher's got a big thesis to read if you want to know all about it, but this is just one snippet of what he found that I thought was kind of interesting and you might find of interest. So he says the rachis and the rachilla nodes on the rachis are where the resistance is occurring, this type 2 resistance we call it, when it's spreading up the spike, and that some of the techniques they can use at the synchrotron are kind of useful to look at this. You could look at it in other ways. One of the benefits of the synchrotron is you don't have to destroy the sample. So you can go back and look at the same plant multiple times as opposed to other methods where you'd have to chop the sample up in small bits and analyze it that way. So, kind of an interesting study. So continuing on from that, we've been looking at trying to find new sources of resistance. So we've been collaborating with Axel Diederken and his staff over at the Plant Gene Resources of Canada, just across the parking lot here. And we've screened a lot of their material. So Lipu Wong has now taken an interest in this project. Lipu's a research officer with us, and funding supported by Sask Wheat, ADF, and Western Grains to try and figure out if there's new sources we can use for Fusarium or Headlight resistance. So we started out by screening. Every year we asked for 2,000 or 3,000 samples. I think they have maybe up to 14,000 samples, I understand, and I think we screened most of those now. We focused on about 4,000 of them for a couple of years that looked a bit better, and then we narrowed that down to 800 in the field, and then to be able to manage it in the greenhouse, we're down to about 200 now of the better ones we think we've got. So these are the scores from field assessment. We're looking at Fusarium Headlight Index. So we look at the number of spikes infected and then the severity on the spike. How much has the pathogen spread up the rachis? And so that gives you a measurement. So you can see that of the 400 here, most of them have reasonably low scores, less than 10%. So hopefully closer to 0 the better, but they're all much better than, when we started screening the initial batch, there was lots that just were worth considering. At the moment, we've taken on Mackenzie. She's a graduate student. You might recognize Mackenzie's name. She was used to receive the ADF applications for a few years there. She's also worked in the Provincial Plant Pathology Lab and decided to do a master's with us. So she's been looking at the FDK. So she's taken those 400 accessions, some of the samples that we had from the field. We looked at the scores in the field, and now what she's doing is hand sorting them to figure out how many Fusarium damaged kernels in those samples. And she's finding quite a lot that have basically none, a lot that have some, but not too much, less than 1% of the kernels infected. So we've got a pretty good collection, not just based on field scores, but also based on the actual Fusarium damaged kernels in the samples. So what Mackenzie's doing is taking them into the greenhouse now and measuring the type 2 resistance again. So you can see on a nice healthy plant, she infects the plant in the middle of the spike here, hasn't spread up the spike, as opposed to an infected plant. Sorry, this one would have been a check, I guess, would have been uninfected, but resistance, we would hope to find something similar. Here she's infected the spike and it's growing up into the tip. But she's also looking at things like flowering date, plant type, disease severity, try and pick out the best lines that we can so that we can start to figure out if they're worth using them for breeding programs. So she's spending quite a bit of time over there at the moment. She made a couple of nice graphs the other day, just basically frequency graphs. I'm not sure why they describe it as density, but basically this means a lot of the accessions have no symptoms. So that's a good thing for the Fusarium head blight index rating. And a lot of them have no FDK. So hopefully we picked out some really good lines. Now what Mackenzie is going to do, and this is charts fresh off the machine, I guess, or off the analysis. It's a Manhattan plot. So it's not as complicated as it looks, but basically she's taking that data from the field, the index, FHB index, or the FDK that she sorted through. And then these lines have been sequenced. So you get all of these, each dot represents maybe 100 base pair sequence. So a small sequence of DNA. And along the bottom here are all the different chromosomes. So 3 genomes, you have 1A, 1B, and 1D, all the way to 7. So those are all the wheat chromosomes. And then you set an arbitrarily or a line here that you suggest that if it's above this line, if these sequences, these dots are above the line, there should be a high chance that they're, very high chance that they're associated with FDK. So hopefully resistance to FDK. So it looks like on 6A, Mackenzie's got a couple of dots there. It looks like there's a couple of markers then that are associated with resistance on 6A. And so when she was looking at those 200 lines now, she's probably got hopefully something that's telling her some of those lines will have some kind of resistance gene on 6A, possibly over here on 3B and another one on 6D. So hopefully that's, we'll be able to find something novel, not similar sources of resistance that are already in use, but something new. So this will be the basis of Mackenzie's MSc. She's been with us less than a year, so she's got lots of time to continue working on this. Okay, the last part of the talk, and spend a little longer on it, is the fungicide work that Dr. Singh did. He's got one paper in plant disease already, and this is to do with timing. So we worked with Gary Nadowich out at Outlook and Gary Peng and his crew up at Melford. We also tried Scott in Indian Head, but didn't have much disease at Scott the year we were out there, and similar at Indian Head. This is work again funded by Sask Wheat, as well as ADF and Western Grains. So if you look on the Sask Wheat website now, you'll see a chart that looks like this that says BBCH59 is when you might consider starting to spray. That's before you see any anthers on that spike, but the spike has cleared the boot and it's above the flag leaf. So this is mainly the ideal time, 61 to 65. So when you start to see all or the majority of the spikes in the field with pretty much all the anthers out, probably getting late. And this 69, when the anthers are starting to dehiss or turn, it's probably getting too late. So this has been, I think, based on trial and error experience, quite a bit of fungicide work in spring wheat and even winter wheat, but very little wheat work, or none to my knowledge really, of fungicide application in durum wheat. So we focused on durum, and we want to just check and see if that was the case for durum. We also looked at high and low plant populations, because folks are saying that, you know, if you're to get to all the heads uniform in a field, it would be better if the plant population was very high. So I remember Tom and I had a talk about this one time. And, he was saying that that's one of the problems for most growers, of course, is trying to stage the crop. When is it actually the right time? You know, when is 61? When is 65? And how many of the spikes have to be at that stage to make a decision to spray? So we looked at plant population, high and low, and we looked at these different stages of spraying. So here we are with the spike out of the boot, but no anthers yet. It's pretty early. Here's 61. If you look closely, you probably see a couple of anthers in the middle of the head. 65, most of them are there. You can't really see them in this photograph. And then 69, they're starting to dry up. We also did a very late application. He did a very late application at early milk. Because we had read in the literature that the Japanese were having some success spraying very late, like 20 days after anthesis. So about almost 3 weeks after 61, they were doing an application or a second application even, and that was supposedly going to manage the toxin, would help to reduce the amount of toxin in the sample. So we gave that a try, and I'll show you the results here soon. So I just want to remind you, 2016 was quite a wet year. That was the year we had a lot of Fusarium head blight problems in the countryside. 17 and 18 were much drier. So 2016, maybe not so good if you're a wheat grower, but it's kind of a pathologist's dream here when you continue to have moisture through flowering through July. And well, we probably don't need it so much in August, but it was an ideal Fusarium head blight year, but of course not if you're growing wheat. In 2017, we did get fairly high infection, but we really didn't have the conditions where it spread up and down the spike. It got warm and dry fairly quickly after flowering. And although we did have some results, they weren't as severe. And 2018, you remember, was quite dry, I think, and not nearly the problems we had 2 years previously. We really only had one site of interest there, and that was up at Melford. Okay, first talk a little bit about the effective seeding rate. What we found, really, our recommendations in terms of timing really didn't depend on seeding rate. They were the same for seeding rate. But there were differences between seeding rates. Obviously, when you seed higher, you get a higher yield generally, as long as you don't run out of moisture and nitrogen. So you can see there's probably about an extra 15 bushels an acre here by going with 400 plants per square meter instead of 75 or so. More interesting is the effect it had on the FDK or DON. So to reduce the amount of FDK in the sample, higher seeding rate helped. Certainly not a cure-all. It is not great. It's not a huge decrease, but there was less FDK at the high seeding rate. You can see it's still very high. It's still over 10%. These are Saskatoon and Outlook sites. Remember, the weather conditions were very conducive to FHB that year. These sites were inoculated and irrigated. So we kind of overdid it, but it did give us a very good look at what happens when FHB is severe. And so we do see a marginal decrease in FDK, and the same thing with DON. So a little less DON at the higher seeding rate. And probably what's happening here is that at the low seeding rate, you've got a lot of secondary tillers. They're not receiving the fungicide at the right time or at all. So you have a lot more variability in the field. So that results probably in a little more damage to the kernels and a little higher toxin levels. So that's the conclusion I think that Grissie came up with, that the high plant population did flower more uniformly. And the nicest thing, or the best thing for him, was it made timing the decision much easier. So low plant population, those spikes are all over, from not emerged to already most of the anthers on the spikes. And so it made it much harder to decide when to spray. And there was, with the higher seeding rate, you had a little higher yield, obviously, quite a bit higher yield, and a marginal or slight reduction in FDK and DON. Optimally timed fungicide applications, similar effect at both high and low seeding rates. So we didn't have an interaction. We wouldn't make different recommendations based on seeding rate as to the timing of the fungicide application. Okay, here I think is the more interesting results. So this is the actual timing. So we're looking again. Remember the chart, 59, so the spike is just clearing the boot here, no anthers yet. You start to see the first few anthers at 61, 65, most of those spikes in the field should have lots of anthers. 69, they're already falling off, they're drying up. and 73 is early milk, so very late. But what we found that was quite interesting, I think, is not a lot of difference in yield between 61 and 65, statistically no difference. Even between 69 and 61, certainly there's trend to a little bit lower at 69, but statistically we can't really say that. It looks similar. So it looks to us that we're getting very similar results in terms of yield right from 61 to 69. And the double application, the two applications, one at 61 and 73, really was no better. In fact, we got better results by one timely application at 65. What was quite interesting, though, was a couple things. One is our unsprayed check here, so no fungicide went on. No difference. So really spraying too early for durum in Gerseeb's plots under very severe conditions really was no benefit to that early application of fungicide. We also included a sprayed check, I'll call it. We are certainly not telling people to do this. It's our attempt at getting a disease-free check. So we sprayed 5 times. So these plots had applications at all the stages. And you can see that even with 5 applications of fungicide, statistically, it's no better than one well-timed application at 65, right? Maybe the trend there to slightly higher yield, but basically they're both got an A on them, which means they're not statistically different. Okay, that's yield. Now let's look at FDK. So you're getting paid on FDK or lack of FDK. How does that compare? So again, what we found was somewhat similar, 61 to 69, statistically no difference. So the trend is downward, but statistically, we're getting the same amount of FDK at 61 to 69. Okay, here the double application may be a marginal difference, a little bit less FDK than say it's at 65, but probably not worthwhile economically to spray twice for that reason. Again, the unsprayed check and the early, before you see any anther stage, no statistical difference. So it doesn't look like that's a very economic treatment for durum based on Gerseeb's results. And then our sprayed check with so much fungicide, we still had, not quite 10% FDK, but it's still quite high. So even our attempt at a disease-free check just doesn't happen no matter how much fungicide you put on. So. Dawn, how much dawn in the sample? Very similar to FDK, 61 to 69. In fact, even the double application, all the same in terms statistically. Looks like a slight trend reduction as you go later. But the dual application, we just didn't see the same results that the Japanese folks got, where they actually saw quite significant reduction in yields by spraying twice. So we think that in most respects, the window of application really is from when you see those first couple of anthers until those anthers are starting to dry up and fall off. And again, we don't see much benefit to that early application. So going too early looks of no value compared to the unsprayed check. So under high disease pressure, growers might consider extending their window of application as late as 69. So going from 61 to 69 might be useful. There's often reasons you can't spray right at 61 or 69. Just you have so much to spray for one thing. Might be raining at that time. So this might give you an extra couple of days to consider spraying. That 69 maybe shouldn't be considered too late. And 59, we need to do more work on this, but we're wondering if that's really not a bit too early to be suggesting to spray. And the dual application, unfortunately, we just didn't see the benefit to the dual application. We didn't get a huge reduction in toxin. Certainly it was lower, but probably not low enough to make it economic. So we were talking about this the other day. We're wondering, is this too early? Is 59 too early? It's from Gerseeb's results on durum, it looks like it. And at this end of the window, it looks like 69 maybe isn't too late. Maybe a slight reduction in yield, but you also get a reduction in toxin and FDK. Okay, last thing I want to touch on. We've been spraying fungicide a lot now for the last 10 years for Fusarium head blight. In other countries, in New York State, for instance, we had the external examiner from New York on Gerseeb's defense last month. they are finding that fungicides are less effective now than they were 20 years ago for managing Fusarium headlight. So Gersebe looked at this. He had a number of isolates, 250 isolates of Fusarium graminearum over a different time period. So from, I think he had some maybe as early as the 1980s, 90s, right up to the present time. He had them from across the prairies, from Alberta to Manitoba. And he had different chemotypes, the 15 and 3-ADON chemotypes in particular. So we're a bit concerned that, you know, our fungicides could lose their effectiveness with the amount of fungicide being sprayed these days. They're single site. We're using the triazoles pretty much exclusively for Fusarium head blight. So there's 3 popular actives, but they're all triazoles. We've got a diverse fungal population, Fusarium graminearum, and most of the Fusariums, there's a sexual stage. So you have sexual recombination, genetic variability, high mutation rate. That allows the fungus to perhaps adapt to the fungicide. And those spores will move, those ascospores in particular, will move some distance by wind every year. So they can spread fairly rapidly. And as I said, fairly frequent application of fungicide these days. So we did this project, or Gersebe did this project in collaboration with Pierre Faubert and Isan Seri over at National Research Council. This was part of a project that they had with ADF and Sask Wheat. And Gersebe's part was to look at the fungicide resistance aspect. So we started by using some of those same samples he had analyzed from 2014 to 16, but he also enlarged it. Like I said, he had samples from Alberta, Manitoba, and over quite a wide range of years. First of all, he made sure they were graminearum by growing them out on plates here. You can see the nice pink color is graminearum. The white one's probably a different species. And then he confirmed it with molecular identification. So he looked at about, I think it was 260 isolates in total. And he looked at the 3 most popular actives. So tebuconazole and prothioconazole, if you put those together, you've got Prozero. Prothioconazole is proline, I think, on its own, where metconazole on its own is caramba. So if I'm not mistaken, I think caramba and Prozero is still the most popular products used. So he looked at those over a range of concentrations. So I'll just tell you a bit about how he did it. These are little plates. They're about, I don't know, 3 inches wide by 4 inches long, and you have all these little wells in each plate. So in each well, you can put about 1/5 of a milliliter, right? About 200 microliters. So you put your media in there, and then you inoculate with your fungus. So he did 3-ADON types, he did 15-ADON types, he did some from Alberta, Manitoba. And you can see when there's no fungicide in this one, this top row, there's no fungicide, that's our check. You can see the fungus growing, that's kind of the color is kind of brown there. You start to put increasing rates of fungicide in each row, you can see less growth of the fungus. And so you put it in this fancy machine here, the bottom right, and what it does is shine the light through it and you measure the absorbance. So obviously when there's a lot of fungal growth, you're going to have a lot more absorbance. When you got a lot of fungicide down here, there's basically no fungal growth, you'll have less absorbance. The light will transmit right through the plate. So then what he did is look at the EC50 value. So that's the effective concentration. So what concentration of fungicide, right? Which row do you look at that has only about 50% of the growth of the control? The top row is the control, right? No fungicide. And then you express the data that way. So I'm happy to tell you, we did not see anything we believe is resistant to the fungicide, insensitive to the fungicide. So I didn't put all 250 isolates, but there's about 40 along the bottom here. These are all the isolates of the pathogen. And this line represents the average of the 250 isolates that Gersebe examined. You can see the score is about 0.03 micrograms per liter of the chemical here, right? So that's the average response of all these isolates. Usually it's somewhat subjective, but usually to say that an isolate is insensitive or resistant to the fungicide, you'd want to see at least a factor of 10. I've read some people don't believe it's resistant till they see a factor of 100. So you can see here at 0.03, we're not even getting double 0.06, right? So this one isolate here on the left. It's a little more tolerant to the fungicide than others. This one down here, less tolerant, but that's just natural variability, we feel. So we don't think any of these isolates are insensitive to prothioconazole, as has been shown. Well, not sure what prothioconazole, but some fungicides have been shown to be less effective after years of use in certain areas of the world. Tebuconazole, the data is a bit more variable, but again, only in, the variability is only maybe by a factor of 2 or 3. If it's 0.07 here, we're getting to about 0.15, so it's only about double. It's nowhere near 10 or 100 times that we're seeing the EC50 that we would consider it then that maybe we've got isolates that are resistant to the fungicide. And the same thing with metconazole or caramba. Basically natural variability here, but nothing that worries us yet that fungicides are not working, that the fungi are becoming insensitive. So I think we're pleased with that so far that's good. It's just we need to keep an eye on this because certainly we have seen erosion in other parts of the world. So we tested those 3 common triazole fungicides for suppression of FHB, and we do not believe that we have any that are resistant to the fungicides at this point. And that's based on isolates, we looked at for the last 20 years across the 3 prairie provinces. So what can growers do to reduce the risk? Start with clean seed, possibly even a seed treatment to be sure that you're getting the crop off to a good start. Follow a diverse rotation. I firmly believe the more diverse the rotation, the better off we are generally. Unfortunately, we don't have resistant varieties in all wheat classes, but the CWRS, the hard reds, are getting pretty good. In a severe year like 2016, I'm sure you'll still find Fusarium, Headlight, but maybe not as bad as you would have 20 years ago. Scout for the disease and apply based on risk rather than routine application. So, I hear quite a few growers say, I just spray pretty much every year now. I don't think too hard about it, but that does increase the risk that the fungicide will become ineffective if we're using them continuously. And only a recommended number of treatments, which so far I think is 1 for most growers. So that's my very brief summary of some of the work we're doing in the lab. Just end off with a quick summary, I guess, that I think an integrated approach to manage this disease is the best thing to do, probably the most economically beneficial and stable. Unfortunately, we can not spray our way out of it. Fungicides are one tool, but they're all for suppression. Nobody's saying you're going to control the disease, even with a well-timed fungicide. You need to couple that with a better variety, or as good a variety as you can get for resistance, and hopefully use a reasonably diverse crop rotation to manage the disease. Timely application will improve things as opposed to, say, spraying too early or too late, but it's really suppression, it's not control. Upping the seeding rate might be beneficial, especially for crop staging, and hopefully getting most of that fungicide on most of those heads when they're at the critical stage. And we haven't seen evidence of fungicide insensitivity yet, but we'll be on the lookout to make sure that we'll let you know if things change. So combine a diverse rotation, resistant cultivars, and fungicide with when warranted, most efficacious way to deal with the disease. So I think, Dallas, I kept pretty good time. I think I'm about 45 minutes or so, and I maybe went a little fast in places, but I'm happy to take.
Okay, nobody has a question yet here. Unless somebody puts up their hand really quick, I'm going to, here's one that just came in. Okay. Maria, your student here, she said, awesome presentation, Randy. AFC London found out a new toxin transforming enzyme. Do you think that's a reality to become true here in Canada?
No idea. It'd be interesting. How would it be? You'd have to know how it was applied. Would it be applied like a fungicide, or would it be something you'd breed into the crop? I mean, I think I've read about that, but wasn't quite clear how they would make use of it. But certainly if they could detoxify things, but you still have, would you still have the FDK? I mean, the toxins are involved in the infection process, so you would think you should also have less FDK as well. But to my knowledge, most grain is bought on visual appearance, right? Unless you're taking it to the field lot or something where they do a toxin analysis directly. But the sample also has to look good. And sometimes the amount of toxin and the number of FDK are not as highly correlated as maybe we'd hope. That sometimes you can have a sample that doesn't look that great and yet doesn't have that much toxin. And other times the exact opposite, where the sample doesn't look bad, yet it does have quite a bit of toxin. But anyway, certainly getting rid of the toxin would be an improvement, of course. Yeah.
Great. One more question that just came in. Aside from fungicides, how is research advancing with biologics to control FHB? Sorry.
There are people working on it. I don't know if you remember, there was an article in Western Producer maybe 6 months or a year ago. My colleague Alan Shu, who's in Ottawa, had found a biocontrol agent, 25, 30 years ago. I don't think it's on the market yet, right? So it's unfortunate, but I don't think a lot of options yet. But certainly there are people working in that area. I hope someday we have things that can compete with, tebuconazole and metconazole, but right now, I don't think so.
Okay. One more question here from Wayne Truman. Thanks for the presentation, Randy. What are the pros and cons of spraying fungicide to protect the flag leaf in comparison to head timing?
Well, flag leaf, you're really just after the flag leaf spotting diseases, right? So septorias, tan spots, spot blotch. If those are big issues, you can have a reasonable yield increase. If you have a wet, kind of a damp, humid, warm year, on the eastern side of Saskatchewan, we tend to see more spot blotch. It likes it warmer, and as you go west, we might see less spot blotch, but they're quite effective. And we've shown, we had a student a few years ago that showed even without Fusarium head blight, even though we timed it for Fusarium head blight, we didn't have a lot of Fusarium head blight, and yet we still had some response because we were controlling the leaf spots. So the leaf spots were not as well controlled as if we had applied at flag leaf timing, but they were still controlled when we applied at anthesis. So, it's a week later or so. So we did improve the sample a bit. We had better 1,000 kernel weight, better test weight. But basically, then you're not spraying. If you're spraying at the flag leaf timing for leaf spots, you're not spraying for Fusarium. It looks like to us that anything pre-59, right, before the spikes out of the boot, you're not going to have any impact on Fusarium. So you have to make the decision. If you think Fusarium is not going to be a problem, and yet leaf spots are, then yeah, I would suggest applying at flag leaf timing. But if you're worried about Fusarium, shoot for Fusarium. You'll still get some leaf spot control, pretty good leaf spot control probably.